PCR, or polymerase chain reaction, is an extraordinary tool which has revolutionized the way we study DNA. The goal here is to understand its significance, explore how it works, and examine its social impact. What is PCR technology? PCR is a technology that amplifies and copies small components of DNA, often producing millions to billions of a specific DNA sequence. Now, this is essential for molecular and genetic analysis, as well as for studying isolated pieces of DNA. In fact, PCR is so prevalent in molecular biology that its creator, Carey B. Mullis, was given the Nobel Prize for Chemistry in 1993. Great, so how does it work? It begins with a double helix, the building block of DNA. Now imagine that we twist this double helix and heat it up to around 94 to 98 degrees Celsius. This will separate its double strands into pairs of singular strands by breaking the hydrogen bonds within the nucleotide base pairs. Next, we go to the second phase, annealing, where the strands are cooled down to 50 to 65 degrees to allow primers to attach onto specific locations. Each primer serves a map for the polymerase. Imagine placing tiny markers at certain points in the maze, guiding the polymerase to the right areas. In the final step of polymerase chain reaction, extension, we raise the temperature to approximately 72 degrees. The goal here is to get DNA polymerase to synthesize a new DNA strand, and this temperature is optimal for Taq polymerase, a heat-stable enzyme derived from Thermus aquaticus. The DNA polymerase moves along a complete strand, matching complementary traces thymine with adenine and guanine with cytosine. As you'll see, the nucleotides will move up the extended DNA strands.
This process is repeated in cycles, the DNA replicating exponentially with each cycle, going from 1 to 2 and then to 4 copies. Generally, it will be in a consecutive power of 2. Therefore, this will give the form 2 to the power of n copies after n cycles, where n is the number of cycles. After 30 cycles, more than 1,000,000 copies are made from a single molecule. But how did this idea come to fruition? It wasn't just the work of Carey Mullis, but rather the result of peer review and collaboration, leading to improved versions and new applications. As you can see, PCR was first created in 1983. However, just a decade later in 1993, the first FIDA-approved kit came to market. Another decade later in 2003, quantitative PCR was discovered. This reduced detection time and used a lower concentration of targeted RNA and DNA samples. Then, in 2020, PCR was used for COVID testing. First, healthcare workers collect samples via a saliva tube or nasal swab. COVID's pathogen, SARS-CoV-2, uses RNA in its genetic makeup. PCR converts itself from single-stranded RNA to double-stranded DNA through reverse transcription. And then the two strands separate with primers attaching to each one. After they're attached, new strands of DNA extend along the initial template strand using fluorescent dyes in the process. This serves as a signal for successful replication, and the process repeats to create millions of copies of the SARS-CoV-2 RNA. If there's viral RNA combined with primers and dye, then the result will be positive. If there's a mismatch and no amplification, then a negative result occurs, meaning that sample didn't contain the SARS-CoV-2 pathogen. This significant development and usage of PCR throughout history undermines the importance of peer review and collaboration in scientific adaptation. Next, we'll delve into the roots of what makes PCR so valuable to the intertwined relationship between science and society, and how it amplifies our understanding of DNA. Imagine you had a way to amplify a tiny sample of DNA to billions of copies. Now, not only would this be useful for analyzing pathogens, but can also be used to identify murders and DNA profiling, as well as studying ancient fossils. These use cases are proven to be extraordinarily relevant in fields of forensics, medicine, and environmental studies. In forensics, PCR is used for DNA profiling. Even a droplet of blood can be used to trace culprit and find missing people. As mentioned earlier, PCR has made waves in testing viruses such as COVID-19 by detecting pathogens, diagnosing diseases through reverse transcription, as well as monitoring the effectiveness of treatments. This extends to tracing rare and unique species of organisms, as well as monitoring ecosystems by detecting water contamination levels, and also tracking endangered populations. Ethical consideration may breach privacy of genetic information, give access and equity in technology, and regulate biotechnology. To conclude, PCR is a revolutionizing technology with an astounding impact on both science and society, made possible through meticulous peer review and collaboration throughout the global community. It democratized biological research, creating a common language for global science, and successfully laid a path between science and society.